Thank you for having me today. I want to thank ARM for organizing always such great meetings and an opportunity to discuss with our peers about what we are developing. I don't need to explain how successful the sector has been in the past decade and how we have really pushed boundaries in science. But at NKILT, we believe that it is time to also look at things differently and try to really bring other approaches to cell therapy if we want to solve the challenge of solid tumor and other hematologic malignancies beyond what has been done brilliantly with CD19 and BCMA. And we also believe that it is time to focus on finding more affordable and accessible to patients by going through the allogenic and building true off-the-shelf technology. So that's why with my co-founders that have significant experience and expertise in cell therapy, but also have some prior biotech experience, we decided to create NKILT with the main goal of bringing the concept of natural receptor to cell therapy. We want to build an off-the-shelf technology. As I said, it is truly important for us to ensure that we have more chances of having patients accessing our therapy. And despite the tremendous efforts made with autologous therapy, it still remains highly costly, very tedious manufacturing process that are limiting the potential access for patients. We believe that through our proprietary chimeric ILT receptor binding technology, we can build a contributor to the next generation of cell therapy and hope to implement in the clinic our first allogenic gene-modified CURE-NK cells focusing on the targeting of HLA-G. HLA-G is a very interesting target that is complex, unique, and only expressed in humans and a handful of primates. That makes it probably more difficult to study. That being said, it is known to be the molecule of pregnancy that is used by the fetus to neutralize the immune system of the mother and ensuring that it will not kill the fetus. It does that through the ILT receptors that are neutralizing, and it's a potent inhibitory receptor, neutralizing all immune cells, making them unable to recognize the fetus and ignore it. While in cancer, over half of cancer cells are expressing HLA-G using that exact same mechanism that is allowing cancer cells to completely shut down the immune system and making the immune cells unable to recognize the cancer cells. The other benefit of HLA-G is that it's solely expressed during pregnancy in placenta cells, and it's barely expressed in any other type of cells, making it a good candidate to limit on-target off-tumor effect. It is also known, as I said, it's only expressed in few mammals, like a handful of primates and human beings. It's a very exquisite tumor antigen. So we believe our technology will affect cancer in 4 different ways. The expression of HLA-G across cancer being large and having a lot of potential for us. Our key specificities and unique properties would differentiate from a standard CAR. I'll go to that in a minute. The way that we believe we can activate our NK cells to make them active in vivo much longer than the standard NK cells. And some words about the tumor vulnerability and how HLA-G and tumor microenvironment have a tight interaction that are reinforcing each other, making the tumor microenvironment even more immunosuppressive, and how we believe our cells can affect that. So regarding the expression, HLA-G has been shown to be expressed pretty broadly across multiple cancers. You can see here some examples which we admit the literature is a little old, and we will be contributing to eventually update those. We need to also understand better the way it is expressed, the heterogeneity of the expression, because it's one of the components that is important to look at. But we think the field is unfortunately wide enough, and you can see some tumors here, whether it's AML that we decided to focus first as a proof of concept, or rapidly going into solid tumor where you can see some high expression, making those cancer that still need new therapies because patients still relapse a good target for us to focus. The other key component about our technology where we think we can make a difference is that, maybe people say, why are you not using a classic antibody or a CAR construct? Well, one of the issues of HLA-G is that it is actually coming in 7 different isoforms. Each isoform are having significant differences, as you can see on the cartoon on the right. Those isoforms have structural differences that will make any form of antibody that you would target against HLA-G1, for example, that is the most expressed, unable to really work on any other isoform. We know that HLA-G is extensively involved in tumor escape mechanism. One of the reasons could be this virus secretion of different isoforms, including soluble forms that will make an antibody facing limitation at some point when they will have eradicated the one they can target. We believe that our approach, not using a standard ACFV, but a protein fusion binding through ILT, is going to address and help improving there. So the main characteristic of HLA-G is being different from all the other HLAs. All the HLAs are, providing that positive signal, while HLA-G is a true inhibitor triggering immunosuppression. And we kind of like, HLA-G is kind of like making the immune system unable to really act against the cancer cells. And the reason why we wanted to use a natural receptor binding is because we thought that copying what nature is doing best is the best way to have an impact. And we also have a specific proprietary activation signaling that is part of our construct that unfortunately I cannot detail because the IP is not completely yet finalized and we're not going to share too much information about it. But it will allow us to have our NK cells staying active for up to 6 weeks, at least at first, maybe more, which is significantly more than what classic and standard NK cells will have, which is on average 2 weeks. Last point about the tumor microenvironment is that there are some really interesting interactions between HLA-G and the tumor microenvironment. We know that many factors of the TME are activating HLA-G expression in tumor cells, like stimulating it, which will increase that immunosuppressive effect around the tumor. We also know that some cells are literally having I would call that an auto-activation, or actually a feedback between themselves that will continue to lead to tumor escape mechanism. And we think that we can contribute in neutralizing this. And the idea of using our cure technology that will not only target HLA-G that is expressed on the tumor, but also target the soluble form that is in the tumor microenvironment will help killing cancer cells, but also neutralizing this blanket of immunosuppressive effect that is not helping other potential treatment. And even if we are not maybe able to eradicate all cancer cells, at least we can limit this immunosuppressive effect and make the tumor more vulnerable. So as I said, these are 4 ways we believe we can affect cancer, and we are very pleased to share some of our preliminary in vitro data that are confirming that our cure construct has been working in T cells and NK cells, and I'm going to run through some quick data for you to understand how we did this. Our initial effort was to focus on validating the cure concept first. It's a new construct. We want to be sure that it is working properly and it doesn't have any issues. Then we were working on optimizing the different type of cure clones that you can see highlighted here with different numbers, whether it was on ILT2 or ILT4 binding approach. We decided to look first at the cytotoxicity of our cells, what we will call cure T cells, based on T cells first, against AML cell line that are expressing low to no HLA-G naturally, but were forced to express HLA-G1. And you can see that a couple of our construct, like the KIR3 on the left side, were showing pretty significant impact on cancer cells. We used the CD33 CAR T that is derived from the same healthy donors as a control, knowing that a CD33 CAR is known to be pretty strong at killing leukemia cells. So this first experiment on T cells was really a way for us to kind of say, okay, it does work, it's transducing, it's stable, it's killing cancer cells, but this is not enough. So we decided to test also our cytotoxicity of our KIR T cells against other cell lines like MOLM14. Here you can see that some of our KIR constructs were pretty good killers, like KIR5, but we can also see that a standard HLA-G CAR was not necessarily performing as well against this cell. We had another experiment not shown here that are confirming that these HLA-G CAR could actually target some other AML cell lines and be very efficient. So we tried to dig a little further and see where was that coming from. And we tested the idea that the HLA-G isoform specificity could be responsible for this. And what we did is that we actually looked at two types of cell lines that were expressing either HLA-G1 or HLA-G2, and you could see that our KIR will be facing similar impact on both expression, while the HLA-G CAR will be better on one versus another, showing that this potential specificity is becoming an issue for the CAR. We really feel that the versatility of our KIR binders to recognize HLA-G isoform is a real asset of our technology that will allow us to eventually overperform antibodies or standard CAR construct with that target. So I keep showing data with T cells, and the company is called NKILT. So when are we going to work on NK cells? Well, the number one reason first why we didn't go to NK cell first, it's because the innate activity of NK cell could obscure the overall efficacy. And if we would not be really able to say if it's really coming from the cells, from the construct, or from any other reason. That's why we went for T cells that were much easier to isolate. But here we showed the innate activity of our cells in the experiment. On the left panel, you can see human peripheral blood NK cells efficiently and stably stranduous with our KIR construct. In co-culture with Kazumi-1 AML cells, we show that even the non-transduced NK cells, as expected, because they are good at killing leukemia cells, are showing some control of tumor cell expansion. But you can see here that our KIR3 NK cells are really having a nicer and enhanced impact over 7 days, and it didn't have any effect on variability despite having different healthy donors. We really believe that also with the fact that they're a good producer of cytokine that can inflame the tumor, and particularly the tumor microenvironment, we believe that this second generation of KIR NK construct are designed to enhance both killing and persistence of our KIR NK cells. So last but not least, highlighting some of the latest data that we have here is that we tested the previous result by co-culturing in other AML cells at lower effector to target ratio, which was important. We did here, it's a 1-to-5 ratio. And bottom line, the efficacy is still there. The cancer killing is still there. I didn't have time to update these slides, but I have from the lab just before the Easter weekend, we did the same experiment on a 1-to-10 ratio, and the data is actually even better, which was a nice surprise. And the team is working on a 1-to-20 study currently, which we believe is actually leading to the same direction, which was really important for us to be sure that we would have our cells being true killers and not just because they are in numbers, so they're going to kill leukemia cells. We needed to be in an approach that will be more back to what will be the reality of treating. So we are very encouraged with those data, and we actually, based on this primary proof of concept data, we are raising $1 to $2 million currently, and I should say urgently, because the science is running fast and we are literally need more studies. But we have a very detailed plan that I highlighted here, where we want to not only continue to fine-tune our construct with the goal of naming our lead assets by end of the year, initiate our process development, of course, in the second half of this year, and eventually start our solid tumor program, where we want to engage our NK cells against numerous different types of solid tumor to see if they can continue to play the game the way they are doing. No, I'm about to finish. I want to just take 30 seconds to thank the people that we support in our activities that are highlighted here. But I also want to have a word for a friend of mine that is, for me, a reminder of why we, NKILT, and we as a community are doing this. I promise that I will always think about him. His name is Oggy. He has been diagnosed 2.5 years with stage 4 pancreatic cancer. He was doing super well. He's a musician. He used to play every week. And he decided 2 weeks ago to stop because he was in a clinical trial and it was not going well. The cancer is taking over and he's losing his battle to cancer. For me, when someone like Oggy decides to go home and not receive any treatment, we are just like seeing the limit of what's happening currently. We need to continue to find solutions. What we are doing at NKILT, what everybody is doing in this room and in this conference, is for people like Oggy, and I want us to be really proud of what we're doing, but never forget that we have to do better. Thank you for your attention.